Chronic wasting disease, prevention, management, and awareness. Devastating diseases come in all varieties, attacking many different species. To name a few, scabies often attacks sheep, distemper attacks both felines and canines. Rabies affects many different species, including deer and raccoons. White nose syndrome is an increasingly devastating disease in bat populations. And mad cow disease has impacted cattle herd integrity for decades.
CWD is what we refer to it as, the acronym. It's a brain disease that affects cervids. So we're talking about white-tailed deer, mule deer, moose, elk. They can all get CWD. They've all gotten CWD. Obviously, we don't have elk or elk or mule deer or moose here in Delaware, but the western states have found it out there. It's a brain disease, neurological disease that affects the central nervous system of cervids. And it is if an animal contracts it, it's 100% fatal. Those animals will die. And as the name suggests, wasting, generally the animal gets infected, and over a period of time, basically its body condition deteriorates and wastes away, ultimately leading to its death. We have two. One being if CWD is found in a neighboring state and it's slowly spreading, and so it's inevitable that it's eventually going to come into Delaware. You know, we're not going to build a fence out of Delaware when it's coming into here. That would have a different response as opposed to if we just found it in the middle of Delaware. If we found it in the middle of Delaware, we try and be more aggressive with trying to eliminate the disease through reducing the deer population, to try and, you know, the idea being fewer deer in that area, less chance of individuals coming in contact with one another and spreading the disease. Whereas if we had this gradual bleed in where it's just inevitably going to be here, we would probably try and reduce the deer population just to reduce its spreading rate, recognizing that we're not likely to get rid of it at that point. Maryland is the closest state that has found CWD, and while, you know, we border Maryland, it's roughly 150 miles away as crow flies.
Okay.
It's out in western Maryland in Allegheny County.
Okay.
So while we're bordering a state with it, it's not that close to Delaware, at least where it's been found so far. Hopefully to never find it.
Yeah.
Our goal right now is to collect 600 samples each year, so that translates to 200 in each county. We sample pretty intensively because our hope is, you know, the more samples you collect, the greater likelihood you are to find the disease, and in turn, the greater likelihood you'd find it early on of it, you know, getting here. As of if you're only collecting 2 samples every year, by the time you'd maybe ever find it, it'd be full-blown, you know, really high infection rate. So our goal is to sample really intensively so that if we do ever find it, it'll be early on from it first getting here, and then hopefully we can, you know, eradicate it by removing those infected animals and stuff. So that's our goal and our long-range plan right now. Both directly and indirectly. So what that means, animals can the disease can be spread from direct contact from an animal, whether it's a doe grooming a fawn, or you know, another animal, you know, licking each other, you know, bucks with does. It can also be spread indirectly. They've taken captive pens where animals were housed that had CWD, removed those animals, did everything they could to clean it, you know, try and, you know, do the best you could to say, all right, this pen would be free of CWD. Put new animals in, and those animals contract the disease. So these prions can be left behind, but then another animal can come through there and pick up those things, and and then pick up the disease, so it can be spread both ways. First found out west.
Okay.
No, was it a spontaneous mutation? It's in the same family of diseases as mad cow,
okay,
scrapie, Creutzfeldt-Jakob disease in humans. So was it a just freak mutation of scrapie or something like that? They're not exactly sure how it, originated, but it was first found in mule deer out west, I think in Wyoming.
Okay. And
that was back in the 70s, I believe, too. I mean, there's a lot that's not understood about it.
Mm-hmm.
Obviously, if you're going to, to do stuff like that, you have to be sampling enough animals, to find prevalence. And one of the problems with it is this disease has a long incubatory period. So you could have an animal that has CWD, you test it, and it came back negative, but that may have been just contracted CWD. In areas where it's widespread, it's common, it's been there for years, they are seeing some population declines and stuff with the herds out there. Particularly if the management goals were for older age herds. If the disease, say, takes 2 to 3 years to kill an animal, certainly that's going to vary from animal to animal. And you're trying to manage an elk population to get 6, 7, 8-year-old bulls, but you have a really high CWD infection rate, it's going to prevent you from getting some of those animals to older age classes because they're going to die beforehand. We don't do the sampling.
Okay.
They go to a lab in Pennsylvania where the test is actually done. Basically, they take a really thin cross-section of the tissues, apply a dye to it, and look at it through a microscope, and if it has CWD, there are pink blotches that'll show up. I guess whatever chemical they use or whatever magnifies the prions that are in there that are the CWD agent. And then hopefully at the end of the year I'll get an Excel spreadsheet back that says no positives for all of them. So we just start out with writing the tag number on the vial, and then based on the data sheet, this is how, with the tag numbers, how we link the data from the hunter to the actual sample. And so I start out by, right here's the voice box. In front of that, make an incision and sort of make an angular cut back right behind, right here's the back of the jawbone. Pull this out of the way and make a cut all the way down to the base of the skull. Pull it out of the way. And clean some of the tissues out of the way. Right here is actually the base of the skull. And here's the very first vertebrae, and what I'm trying to do is cut the tissues and go right in between the first vertebrae and the skull. So I'll pull that out of the way. What we're interested in is the obex. That's the portion of the brain here where it meets the spinal cord. Pull that out. What specifically is the obex is that little Y area right there. So then we'll drop that into the formalin to preserve the tissue. And then also what we want are the retropharyngeal lymph nodes. This is the back of the jawbone. Sort of if you drew a line, diagonal line down to here, they're located right on that line. Here's one of the lymph nodes. I'm just going to basically butterfly it to increase the surface area for the formalin to get in there and preserve the tissue. And then here's the other lymph node on the other side. This is an average size lymph node. Some of them are smaller, some of them are bigger. Normally they're this color, sometimes they're purple. I don't know why they're big, small, different colors, but so that's it. That's a CWD sample for deer 3981.
CWD is a contagious neurological disease that affects the brain tissue of cervix. It is a result of malfunctioning proteins called prions. These misfolded proteins cause other proteins in the body to break down, resulting in a chain reaction that ends in cell death. This cell death causes the holes in the brain. This disease is considered to be almost 100% fatal. CWD prions can be found in the eyeballs, brain, spinal cord, spleen, and lymph nodes. All of these should be avoided when field dressing any species of cervids. Presently, CWD has not been transmitted from cervids to any other animal. However, hunters should still take some precautions when field dressing. Hunters should use a sharp knife and wear rubber gloves to prevent contamination from body fluids. If the weather is cold, it is advisable to wear a cheap pair of disposable knit gloves to reduce the chance of cold, clumsy hands. Please contact your state's Division of Fish and Wildlife for regulations involving the disposal of internal organs after field dressing and the testing of harvested game for CWD before consumption in known CWD areas. The symptoms of CWD include staggering, drooling, excessive thirst and urination, rough coats, an odd wide stance, acting lethargic, and a lack of interest in feeding. Infected animals appear to be slowly wasting away, and this eventually results in the animal's death.